Welcome back, folks, to Chapter 3, Mendelian Genetics or Mendelian Inheritance. I'm going to break this chapter up into two lectures. The first one will be most of the chapter, but then I'm going to do a separate lecture for the statistics, the binomial expansion, the chi-squared. It's just because I have to do a lot of things manually and that part of it, so it's just going to take too long. So there'll be two lectures you'll have here. All right, who was Gregor Mendel? Well, first of all, Gregor Mendel was an Austrian monk who was studying pea plants, Pisum sativum, and he was studying 7 or 8 different characteristics of the pea plants and traits that we'll look at in a second. And this was a time, he did this in the mid 1800s. He was a contemporary of Darwin, although they never talked. Just contemporary of Darwin means he lived around the same time Darwin did his experiments around the same time Darwin was doing his. They weren't in contact with each other. Probably would have been kind of cool if they did, but they didn't. And then Mendel's work. Mendel was a mathematician. In the mid 1800s, we didn't even know that DNA was the genetic material. We didn't start to even be suspicious of that until the early 1900s. And then that wasn't even demonstrated conclusively until 1944. So what we're looking at here is a mathematician that is breeding pea plants, and he came up with some laws of genetics, some genetic laws based on these breeding experiments, based on math, basically. He went into obscurity. Mendel was not well known for his work until the early 1900s when his work was rediscovered, and scientists were seeing how important his work was. So let's talk about Gregor Mendel a little bit. You can see a picture of Gregor Mendel here on the left. You can see the pea plant over here. You can see the pod. You can see the seeds inside. You can see the flower itself right here. We'll talk about axial and dominance and terminal dominance. We'll talk about that. You can see a flower right here. So we'll talk a little bit about the pea plant. Again, this is not a botany class, but we do have to do a little bit of botany just to understand what he's looking at. All right, here's some of the traits that he was looking at. He looked at the characteristic height, and of course, within the characteristic height, you can have a trait of being tall, or you could have a trait of being short. They call it tall and dwarf. Here's another characteristic, flower color, and here's the trait, purple or white. Here's another characteristic, flower position, whether it's axial or terminal. So you can see that right here. So that's flower position. You can see another characteristic, seed color. Now be careful here. Look how seed color yellow and seed color green. As it turns out, the seed color yellow is going to be the dominant trait over seed color green. But then when you look at pod color, with pod color, green is going to be the dominant trait and yellow is going to be the recessive trait. So when you look at this, remember that seed color and pod color aren't following the same. The color that is dominant in seed is not the color that is dominant in the pod. So make sure that you're careful when we're talking about seed color or pod color, that you know if we're talking about the seeds or the pods. And we're also going to look at seed shape, whether it's round or wrinkled. Sometimes we say smooth and wrinkled, just so you know. But what we've done, because we also have a pod shape, is we've actually said round for seed shape. And the round is dominant over wrinkled, so wrinkled would be little r. And then down in pod shape, we've used the word smooth. Although I will tell you that I have seen the word smooth used for seed shape. So just keep that in mind when you're reading different sources, different literature. So be careful of color, because there's a seed color, there's a pod color. Be careful of shape, because there's a seed shape and a pod shape. All right, a little bit about the Pisum sativum. Monoecious means that you have both sexes on one organism. Now, you could have a monoecious plant and have a female flower and a male flower that are distinct and separate, but they have to occur on the same plant. Or you could have a plant that has a flower that has both male and female parts. There's really two ways to be monoecious. But what monoecious means is both sexes on one plant. That's what monoecious means. Dioecious means that one plant is male and another plant is female. That's what dioecious means. Monoecious literally means one house, so both sexes in one house. But remember, there's two ways to be monoecious. Distinct flowers, two distinct flowers on one plant, a female flower and a male flower. Or you could have one flower that's has both male and female parts. As it turns out, pea plant is this type of monoecious plant. One flower has both male and female parts. And we'll look at a picture of that in a second. This is the picture of the pea plant. You can see that here's the male part, the anther and filament. The anther is the top, the filament's here's the filament. That's the male part. Here's the female part, the stigma, style, and ovary. So stigma, style, and ovary is the female part, of course. If you say ovary, you know what I'm talking about, female. And this is one flower, both male and female parts. Now, what Gregor Mendel could do is he could cut off all of these anthers. He could cut these anthers right off. Cut them, cut them, cut them, cut them. So he could make this flower only female before the anthers made pollen, before they were able to self-fertilize, because pea plants can self-fertilize. And then he could dictate or determine or manipulate taking pollen from another flower, from another plant, and fertilize this flower. So that's why this was an ideal plant to use in his breeding experiments. He could control who fertilized whom. Just a little bit of botany here. There's really 3 cells in a pollen grain. So here's a pollen grain. There's 2 sperm and a pollen tube cell. Can't spell pollen, I see. So 2 sperm and a cell that grows into a pollen tube. So here's the pollen tube growing down through. And there's going to be 2 sperm swimming down through that pollen tube. That pollen tube makes it down and around here to the ovum. There's the ovum. One sperm's going to fertilize the egg right here, and the other sperm is going to fertilize the polar nuclei cell. There's a cell in the center called polar nuclei cell. When the sperm fertilizes the polar nuclei cell, that's going to be triploid, and that becomes the endosperm, or the fruit, or the nutrients for the growing pea plant. There's these synergid cells here. There's 2 synergid cells. They just assist in fertilization. In fact, one of them actually transmits the pollen cell to the egg. And there's these antipodal cells up here. They degrade after a while, but they're involved in nutrient control as well. So the most important thing going on is right here. That's the most important thing going on, because that's the sperm fertilizing the egg, and it's going to become your new pea plant. All right. This is one of the reasons why he chose the pea plant. He could go in with a set of scissors and cut off the anthers, so cut off the male parts, in essence turning this flower into a female flower only. And then he could go to another plant someplace else, and he could take the pollen off the anthers with a paintbrush, and he could bring them over and paint them onto the stigma of the stigma. Remember, the stigma, style, and ovary is the female part. So we could paint them on. So he could control mating a purple flower, big P for purple, with a white flower. He could control that. Just so you know, if I put a line over a letter, it means it's small case. That's a throwback from work in blood bank in the clinical laboratory. There are some letters, of course, like S. Kind of hard to know if it's a big S or a little s. They look the same. P is actually not different. Is that a small p or a big P? Well, if I put them side by side, it's easy to tell, but if I put it all by itself, you might not. So I will sometimes write lines over my small case letters. If you see that, you know it's small case. It's just something I'm used to doing from working in the laboratory department blood bank, because you don't want to make a mistake in blood bank. So we're very clear about if we're talking about a dominant or a recessive allele. All right. So there we are with Mendel so far. These are the characteristics that he looked at and the traits. Remember, a variant character is a trait. So these are traits we're seeing here. Tall dwarf is a trait. Purple white's a trait. Axial terminal's a trait. Yellow green's a trait. Round wrinkle's a trait. Green yellow's a trait. Smooth constricted is a trait. All right, so let's go and look at what he was doing. I will tell you that the first thing he did for this parental generation is he mated a true breeding purple flower with a true breeding white flower. What does that mean? Well, what that means is if you're a true breeding purple, it means that, if you're a true breeding purple, it means that you're homozygous for purple. You're big B, big P. And if you're true breeding white, it means that you are homozygous for the white and you are little p, little p. That's what true breeding means. You're homozygous. As it turns out, there's only one way to be white, and that's little p, little p. But there's two ways to be purple. You could be big P, or big P, little p. And the reason being is because purple is dominant over white. Now, Mendel discovered this dominant recessive relationship. And there's other relationships between genes, which we get to in further chapters, like codominance and incomplete dominance and epistasis and things like this. But for this chapter, we're just going to talk about simple dominant recessive relationships. And in fact, just so you know, Mendel discovered other relationships, but he worked on his dominant recessive relationships and published. Actually, he didn't publish much. Yeah, I say he published. He didn't publish to the widespread scientific community at the time, but he did write his results, and they were rediscovered in the early 1900s. He did most of his work on dominant recessive relationships, even though he did recognize and write about other relationships like incomplete dominance and codominance. All right, so let's take a look at Mendel's experiments.
Gregor Mendel conducted experiments to answer one of the fundamental questions of heredity. What are the basic patterns in the transmission of traits from parents to offspring? In this recreation of Mendel's historic experiments, you will have the opportunity to predict the outcome of two types of genetic crosses using a Punnett square. A Punnett square shows the possible combinations that can result when the gametes come together in the moment of fertilization. In this first example, the plants being mated differ in just one trait. One plant is homozygous for the dominant purple flower allele of a gene. The other is homozygous for a recessive white flower allele. To show the mating, we arrange the parent plants along two sides of the square. Note that each parent produces a single type of gamete. The genotype of each gamete produced by one parent, shown on the top, is brought down into each of the squares below it. Likewise, the genotype of each gamete produced by the other parent, shown on the left, is moved into each square to the right. The Punnett square helps us predict the genotypes of the offspring. Using the information provided, predict the phenotypes of the offspring. Drag the correct phenotypes to each square in the grid.
All right, so I'll drag them for you. Right here, this is going to be big P, little p, and that's a purple flower because we know that big P, purple, is dominant over little p. And the only way to be white is be little p, little p. So that's going to be a purple flower. How come it's not pink? Well, because this is not incomplete dominance. This is a dominant recessive relationship. There are gene interactions where you do have red and white giving you pink, but that's not the case for this particular set of characteristic, this set of traits, and this relationship. This one's big P, little p, and that's going to be purple. This one's big P, little p, and that's going to be purple. And this is big P, little p, and that's going to be purple. And where does it say? Oh, right up here. Right up here it says, correct, congratulations, move on. So we're going to move on. So I hope you guys saw that. That's pretty simple. And when we do some monohybrid crosses, hybrid meaning two heterozygotes mating, a big P little p mating with a big P little p, you'll be able to do this some more. But that's where we're at so far.
Notice that all of these offspring have the same flower phenotype as the purple-flowered plants in the parental generation. In the offspring, called the F1 generation, the white phenotype disappears. If we mate these heterozygous plants to each other or allow them to self-pollinate, what kinds of offspring would you expect? From the choices given, select the correct gametes to place on the two sides of the Punnett square.
All right, so let me just remind you before you, I think you guys probably could do this right now, but what happens is if I have a big P, little p individual, what happens during S phase of interphase is I double my DNA, all right? And then what happens during meiosis 1 is I pull apart my homologs. What happens during meiosis 2 is I pull apart my sisters. And those are my four gametes. Two big P gametes and two little p gametes. So what kind of gametes can I make? I can make a gamete that's a big P, and I can make a gamete that's a little p. Those are the two types of gametes I can make. So because I'm self-fertilizing the F1 generation, and F1 stands for first filial generation. That's what it stands for, first filial. F2 generation would be the second filial generation. F3, third filial generation. So you have the parental generation. F1 are the kids, F2 are the grandkids, F3 are the great-grandkids, etc. There's only two types of gametes that can be made here. Where are they going to let me drag from? Or can I type? Oh, right here. So I can have a big P gamete and I can have a little p gamete. And over here I can have a big P gamete and a little p gamete. And then the eggs are going to be fertilized with the sperm. So in here I'm going to be a big P, big P. In here I'm going to be a big P, little p. Does it allow me to do that? Big P? No, it's not allowing me to do that. It's just saying I'm done. All right, let's see what else they're going to make me do here. I hope you can see where the gametes are coming from because I double my DNA, I pull apart homologs, and I pull apart sisters. And that gives me my four gametes.
Using the information provided, predict the genotypes of the offspring. Drag the correct genotypes to each square in the grid.
All right, so this is where I'm going to drag the genotypes. Now, that's genotypes. Genotype is different from phenotype. Phenotype is how you look, and genotype is what your genes are, what your genes look like. So this is going to be a big P, big P. The genotype is big P, big P. Its phenotype is purple. This one's going to be a big P, little p. Its genotype is big P, little p. Its phenotype is purple. This is going to be a big P, little p. This big P and this little p. And this is going to be a little p, little p. So if I did a genotypic ratio, I'm 1 to 2 to 1. I'm 1 homozygous dominant, I'm 2 heterozygotes, and I'm 1 homozygous recessive. That's if I did a genotypic ratio. If I did a phenotypic ratio, I'm 3 purples to 1 white. So there's a difference between a genotypic ratio and a phenotypic ratio.
Using the information provided, predict the phenotypes of the offspring. Drag the correct phenotypes to each square in the grid.
All right, I'll do this for you. Purple, 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 white.
This first series of crosses recaps the result of a mating in which the parent plants differ in a single trait. The parent plants are homozygous for the dominant and recessive alleles. Their F1 offspring are heterozygous and show the dominant purple flower phenotype. The next generation, called the F2 generation, reveals a typical 3-to-1 ratio of phenotypes. The recessive white-flowered phenotype reappears in this generation after having disappeared in the F1 generation. The two series of crosses shown here illustrate a mating of peas in which the parent plants differ in two traits. These traits are round versus wrinkled texture and yellow versus green color. Each cross yields a 3-to-1 ratio of phenotypes in the F2 generation. If the two heterozygous F1 plants were crossed, what genotypes and phenotypes would result? These homozygous parent plants differ in both the R and the Y genes. The round yellow pea has dominant R alleles for a round phenotype and dominant Y alleles for a yellow phenotype. The wrinkled green pea has recessive R alleles for a wrinkled phenotype and recessive Y alleles for a green phenotype. From the choices given, select the correct gametes to place on the two sides of the Punnett square.
Okay, you can only have, there's actually only one type of gamete you can make here. You can be big R, big Y, and you can be big R, big Y. Remember, you're going to separate the homologs, the big R, and then you're going to separate the sisters. So you're going to double the DNA, then separate homologs, and then separate sisters. Now, here's one thing we're assuming here. We're assuming that R and Y are not on the same chromosome. Let me be clear. If R and Y were on the same chromosome, then they're said to be linked. And as it turned out, he showed that these were not linked, that they independently assorted. So they're not linked. They're not on the same chromosome. So this is the only type of gamete you can make over here is a big R, big Y. Now up here, for whether that's the egg or sperm, I don't know which one's female and male here. Let's just say that the green wrinkled is female and the smooth yellow is male. Up here, there's only one type of gamete you can make, and that's a little R, a little Y. All right, so now let's mate them. And what you're going to get in your first filial generation is you're going to get a heterozygote for both traits. So you're going to be big R, big Y, little y. For every single baby, every single F1 is going to be big R, big Y, little y. And then if we self the F1s this time, that's called a dihybrid cross, because you're not only crossing big R and little r with a big R and little r. That means if you self the F1s, I'm talking. But you're also crossing a big Y, little y. What does it want me to do? Just play. That's what it wants me to do.
Using the information provided, predict the phenotypes of the offspring in each square of the grid. Drag your selections from the phenotype choices.
Okay, now if you're big R, then you're round, not wrinkled. And all of these are big R. And if you're big Y, then you are yellow, not green. Because if you're little y, you're green. All of the babies are round and yellow. Every single one of them are round and yellow. They're all heterozygote for round, and they're all heterozygote for yellow, but they're all round and yellow.
In our cross, notice that all of the F1 individuals show the dominant yellow and round phenotypes. The recessive alleles in this generation are present, but the dominant alleles mask the recessive phenotype. If we mate F1 individuals to each other, or allow them to self-pollinate, what kinds of offspring would you expect? Begin by dragging the correct gametes to the two sides of the Punnett square.
All right, so first of all, let's do the top one first. This big R chromosome is going to be separated from the little r chromosome. But how the big R and little r tetrad lines up means that the big R could go with the big Y, or the big R could go with the little y. That's what that means. So what I'm going to say, and this is just kind of random, meaning I just got to make sure I have the right combination in there. The order I do it doesn't matter. But let's put the big R with the big Y. And then let's put the big R with the little y. All right? Now the big R is done. Those are the two possible ways that big R can assort with the Y chromosomes. And the assortment is random and independent. That's why I can get both of these. And likewise, let's do the big Y now. I'm sorry, let's do the little r now. My little r can go with the big Y, and my little r can go with the little y. Now, I didn't have to do it in that order. It's just that I've done these before, and That's always the order I do it in. I'm always going to have the little r, little y down in this box down here. It's just the way I do it. Um, Actually, if you didn't do, I don't think it would say no to you if you did it out of order. Maybe I'll go back and see if you can do it out of order. And by the way, the other parent is going to have the same types of gametes because both parents are heterozygote for both traits. So I'm going to have a big R, big Y gamete here. I'm going to have a big R, little y gamete here. I'm going to have a little r, big Y gamete here. And I'm going to have a little r, little y gamete here. And now all we got to do is mate them. That's all we got to do now.
Using the information provided, predict the phenotypes of the F2 offspring. Drag your selections from the phenotype choices.
Okay, so this genotype is going to be big R, big R, big Y, big Y. Well, big R, if you have a big R, you're round, and if you have a big Y, you're yellow. That has to be that. This one's big R, this one has at least one big R and at least one big Y. Every single thing that has at least one big R and one big Y is going to be round and yellow. This one has a, yep, but this one has a big R and, oh, it's a big Y. All of these are going to be like that because they all have at least one big R and one big Y, at least. All right, here I have at least one big R and one big Y. Here I do not. All right, I have a big R, but I got two little Y's. I'm round but green. Here, yellow. Here, big R, little Y, little Y. So here's also big R, little Y, just like here. All right, this one's big R, and this one's yellow. This one has a big R and a big Y. This one has a little R. This one's wrinkled, but it is yellow. See, it has two little R's wrinkled, but it is yellow because it has a big Y. This one is wrinkled but yellow, has at least one big Y. It has that big Y right there. This one is round and yellow. This one is round and green. It has two little Y's. This one is little r. This one's green. Oh, I'm sorry. It's wrinkled and yellow. It has a big Y. And then down here, I'm finally wrinkled and green. Now, what I want you to notice here is this. If I look at phenotypic ratios, I have 9 round yellows. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellows. I have 3 round greens. 1, 2, 3. 3 round greens. I have 3 wrinkled yellows. 1, 2, 3. And I only have 1 wrinkled green. This is a 9 to 3 to 3 to 1 ratio. 9 to 3 to 3 to 1 ratio. And everything that doesn't look like the parent is called a non-parental type. In fact, all 7 of these are non-parental types, but all 9 of these are parental types. Because what do my parents look like? My parents are round and yellow. So my 9 round yellow ones are parental types. My round green, my wrinkled yellow, and my wrinkled green are non-parental types.
When the genes for 2 traits are considered, the F2 plants have phenotypes in a ratio of 9 to 3 to 3 to 1. 9 are round and yellow, 3 are round and green, 3 are wrinkled and yellow, 1 is wrinkled and green. This phenotype ratio of the F2 generation is typical of a cross in which the parental generation differs in 2 traits that assort independently of each other. According to Mendel's principle of independent assortment, the genes behave independently of each other during meiosis, resulting in this predictable ratio of phenotypes in the offspring. Although our cross involved a difference in 2 traits, we can examine each trait individually to see the typical 3 to 1 ratios of single trait crosses. There are 12 round peas and 4 wrinkled peas, or a ratio of 3 round peas for every wrinkled pea. There are also 12 yellow peas and 4 green peas, or a ratio of 3 yellow peas for every green pea. In the group of yellow peas, there are 12 round peas and 3 wrinkled peas, making a ratio of 3 round peas for every wrinkled pea. And in the group of green peas, there are 3 round peas and 1 wrinkled pea, again making a ratio of 3 round peas for every wrinkled pea. Regardless of the color phenotype, the texture ratios will always be 3 to 1. Similarly, regardless of the texture genotype, the color ratios will always be 3 to 1. These results are typical for traits with a simple dominance-recessive relationship that assort independently.
That last sentence is huge. So what he really did was when he was interrogating just the texture and he was ignoring the color, or is interrogating the color and ignoring the texture, he was really showing you just a simple Punnett square where you had a big R little r crossed with a big R little r. That's all he was doing. And he was saying it's still a 3 to 1 ratio. If you interrogate the color and ignore the texture, you get a 3 to 1 ratio. If you interrogate the I forget which one I just said. If you interrogate the color and ignore the texture, you get a 3 to 1 ratio. If you interrogate the texture and ignore the color, I think I just said that. You know what I mean. Let me say it. Let me try it like this. If you interrogate the color, if you look at the color and ignore texture, you get a 3 to 1 ratio. If you interrogate the texture and ignore color, you get a 3 to 1 ratio. So he's saying that in this dihybrid cross where I get a 9 to 3 to 3 1 ratio, it's really just 2 monohybrid crosses combined. That's all it really is. But how can this be possible? Well, first of all, they're on different chromosomes. R and Y are on different chromosomes. Because if they weren't on different chromosomes, then the big R, you have one of two choices. The big R, big Y is inherited together. And that means the little r, little y is inherited together because they're mutually exclusive. Or, I mean, it could be that the big R, little y is inherited together, which would mean that the little r, big y is inherited together. But if that was the case, you wouldn't have 4 gametes. You would have 2 gametes per parent. You wouldn't have all 4 of these gametes if they were linked. You would only have 2 gametes because they're inherited together. They're on the same chromosome. So what this dihybrid cross tells us, well, first of all, it tells us the fact that a hybrid, a heterozygote, always gives a 3 to 1 ratio from a monohybrid cross. It tells us yellow is dominant over green. And it tells us that round is dominant over wrinkles. But then the dihybrid cross tells us that these color texture alleles are on different chromosomes because you get all possible combinations of gametes, of sperm and eggs. Because if they were on the same chromosome, you would not get all of those 4 gametes. You'd only get 2 of them. So this dihybrid cross led to his law of independent assortment. And he's saying right here that they assort independently. And they sure do if they're on different chromosomes. All right. So the law of independent assortment was shown by I'm actually going to go through these again. There's 2 laws that Mendel came up with. Law of segregation, he said that you separate your homologs. That's all that the law of segregation says. You separate your homologs. So law of segregation says that you separate your homologs. Now, I have to remind you, because you know something Mendel didn't. You know that during meiosis 1, that you pull apart homologs. Mendel didn't know that. All he said was that the unit of heredity separates. And he actually coined the word gene, so you can use the word gene here. But the unit of heredity, I can't spell hereditary. The unit of heredity separates. So you have a yellow gene from your mom and a green gene from your dad. Well, when you make your sperm and eggs, they separate. That's all he's saying in his law of segregation, that the unit of heredity separates. And the unit of heredity equals gene. And he said it's a discrete unit. That's what he said. The Punnett square comes from a guy that worked with Thomas Hunt Morgan. We'll talk about Thomas Hunt Morgan and fruit flies in the next chapter. He Reginald Punnett worked with Thomas Hunt Morgan. I think he was his graduate student actually. So Reginald Punnett came up with the Punnett square, where you put your sperm on top and your eggs on the side, or vice versa. It doesn't matter. It doesn't matter where you put your you could put your eggs on top and the sperm on the side. It really doesn't matter. And he says if you do a Punnett square, then you'll get all the possibilities of mating here, just like that. So that was Reginald Punnett who worked with Thomas Hunt Morgan.
Gregor Mendel's principle of independent assortment states that genes on different chromosomes behave independently of one another in the production of gametes. In other words, gene segregation patterns parallel the patterns of chromosome behavior in meiosis. This parallel is illustrated here for a diploid cell with homologous pairs of chromosomes.
Now, before I play, I'm gonna keep going. I don't remember where the A's are going to go. Let me actually play and see where they go.
The diploid cell has the genotype big A, big B, little b, with the big A and little a gene pair on one chromosome, and Gregor Mendel's principle of independent assortment states that genes on different chromosomes behave independently of one another in the production of gametes. In other words, gene segregation patterns parallel the patterns of chromosome behavior in meiosis. This parallel is illustrated here for a diploid cell with homologous pairs of chromosomes.
Okay. The A, I forgot. I think the A went to the orange. Notice how the A's go to the orange and the B's go to the blue. At least that's what I thought I saw when I just did it a second ago. But realize that the A's and B's are on different chromosomes. Because if they're on the same chromosomes, then you have linked genes, which we'll talk about later. So remember what you're really doing here. When you have AA and BB, and they're on different chromosomes, during S phase of interphase, you double your A's, you double your B's, and then you pull apart homologs. The big A is going to go to one side, and the little a is going to go to the other side, different cells. And where do the big B's go? Well, the big B could go with the big A, but couldn't the big B go with the little a? And the answer is yeah, it could. You could be big B here, but the big B could be over here with the little a's. And then in meiosis 2, you pull apart these sisters. So then in meiosis 2, you're going to have big A, big B, big A, big B, little a, little b, little a, little b. But remember, back in meiosis 1, you didn't have to put the big B's with the big A's. You very well could have pulled apart those homologs to be like this. And then you pull apart sisters. Now you're going to have a sperm or an egg that's big A, little b, and you're going to have a sperm or an egg that's little a, big B, little a, big B. That's what you're going to have. So that's what this video is showing you. It's showing you the law of segregation. We're going to pull apart homologs.
The diploid cell has the genotype big A, big B, little b, with the big A and little a gene pair on one chromosome and the big B and little b gene pair on the other chromosome. The chromosomes duplicate prior to cell division.
Okay, they didn't do it exactly how I did it, but you can see that the A's and B's are on different chromosomes, and that's very important. I see what they're doing. I can't tell which one's longer, but let's call the A's chromosome number 1 and the B's chromosome number 2. Let's say the blue came from your dad and the orange came from your mom. So your mom gave you the big A and your dad gave you the little a. Your mom gave you the big B and your dad gave you the little b. These two are homologs to each other, and these two are homologs to each other. So 2 number 1s and 2 number 2s, if you will. You need to keep going back, and this really is simple stuff, but we add so much so fast that you forget what's really going on. So you have to keep going back and say, this is just is really simply explained by I got 1 chromosome from my mom and 1 chromosome from my dad. And we start adding letters for the genes, but don't forget what's really going on.
In metaphase 1 of meiosis, the 2 pairs of homologous chromosomes align along the metaphase plate. Because each pair of homologs moves independently of the other, 2 different alignments occur, and each alignment is equally likely.
That's just independent assortment. And now remember, Mendel didn't know that you have this random alignment of chromosomes at the metaphase plate of metaphase 1 of meiosis 1. He didn't know that. But he did see the math at the end, and he says, wow, not only do my units of heredity, genes, segregate, but they're also independently assort. How the gene number A, how gene A separates, is independent of how gene B separates, which is independent of how gene C separates, as long as they're on separate chromosomes.
The left cell has the dominant alleles big A and big B aligned in the same plane on the metaphase plate, and the recessive alleles little a and little b are similarly aligned. In the right cell, the big A and little b alleles are aligned in the same plane, as are the little a and big B alleles. During anaphase 1 of meiosis, homologous chromosomes separate and migrate towards the poles. Telophase 1 follows, and 4 cells are produced. Meiosis 1 is complete.
Well, wait a minute. Okay, do not listen to her right there. Let me tell you what she meant. She said to you, if you play it again, at the end of telophase 1, 4 cells are produced. No, they're not. Two cells. But what she's doing for you is showing you the possibilities. You could be this one right here, or you could be this one right here. Because remember, you didn't have to separate the big A with the little b. You could have separated the big A with the big B. So 4 cells aren't produced at the end of meiosis 1. 2 cells are produced at the end of meiosis 1. But so she misspoke. I really didn't notice that the last time I looked at this video. I just caught it now. But it's either these 2 cells or these 2 cells. It's one or the other. They're mutually exclusive. You're not going to have both. Oh, actually, wait a minute. Did I even point them out right? Maybe I should be pointing horizontally there. If the big A, separate homologs. Yeah, I did right, because you have to separate homologs. All right. Yeah, all right. So that's what she meant. You're not making 4 cells like she just said. You're only making 2 cells. But she showed you both possibilities. That's what she showed you. All right. Let's keep going.
This is followed by meiosis 2, during which sister chromatids separate. The end result is different for each alignment.
And now you don't make 8 cells at the end of meiosis 2. You only make 4 cells. You have a total of 4 cells at the end of meiosis 2. But she's showing you both possibilities. You don't have to have a little a with a little b. You could have a little a with a big B. So she's showing you both possibilities. That's all. All right. I hope that she didn't confuse you, or I hope I didn't confuse you. Please go back and play it and listen carefully if it started being confusing, because she misspoke.
The left cell produces 4 gametes, 2 of which are big A, big B, and 2 of which are little a, little b in genotype. The right cell also produces 4 gametes. However, 2 of these are big A, little b, and 2 are little a, big B in genotype. When all the products of meiosis are considered together, equal frequencies of haploid cells occur with the following genotypes: big A, big B, little a, little b, big A, little b, and little a, big B.
Oh, there you go. So as long as there's nothing else going on, which we'll talk about some weird things in the future, you're going to have 1/4 or 25% of all the possibilities in your sperm or eggs. Now you're going to say to me, yeah, but Kiggins, last lecture you told me that the eggs make these polar bodies. Yeah, I understand, but you're still going to have this 25% because, you're only going to have one secondary oocyte left, but it's still a 25% chance that you have that oocyte left. That oocyte could have been the polar body, and the polar body could have been the oocyte. That's what I'm telling you. So there's a 25% chance of having that combination.
Mendel's laws demonstrate how two members of a gene pair separate from each other in gamete formation, principle of segregation, and how one pair of homologous chromosomes assort independent of another pair of homologous chromosomes during the production of gametes, principle of independent assortment.
Okay, that's it. That's the law of segregation. Law of segregation, your homologs separate. That's what it is. All right, let's talk about a couple things here. We're back to a single factor cross, but I have a question for you. If this is a true breeding tall plant, how do you know it's true breeding? Because you could be tall being big T, or you could be tall being big T, little t. Well, what you do is you do a test cross. And what that means is, before he even does the experiment that you see here, before he ever crosses the pollen of the tall with and paints it on the stigma of the dwarf, before he even does that, he breeds this tall plant with a dwarf plant. And he plants, say, 100 seeds. Now think about this. If that tall plant is truly true breeding, he'll always get tall plants. Always. He'll plant 100 seeds and they will all be tall. 100% of them are tall, as you can see here in this Punnett square. However, if by chance that tall plant isn't true breeding and it's a heterozygote and he didn't know it, because that certainly is a tall plant. Big T, little t is a tall plant. But he didn't realize it wasn't true breeding. Then he'll get 50% tall and 50% short, dwarf. And then he'll know, those are dwarf plants. Then he'll know that he doesn't have a true breeder. And he won't use that in this particular experiment. He'll set it aside. He'll take that potted plant and set it aside and say, you know what, I know you're a heterozygote, I'll use you later, because he might need a heterozygote later. So he knew that he had a true breeder because he did test crosses. And by the way, he didn't have to test cross the dwarf plant because there's only one way to be a dwarf plant, and that's little t, little t. So first of all, he bred a true breeding tall with a true breeding dwarf. He knew that all of the babies had to be heterozygotes. All of the F1 generation had to be heterozygotes. Then what he did was he either self-fertilized or cross-fertilized the F1 generation. I mean, it didn't have to be self-fertilization. Let's say he had 1,000 of these F1 plants growing. He planted 1,000 seeds. Well, he knew every single one of these plants was big T, little t. He knew that because he knew he had true breeding parents in the parental generation. So he either selfed them or he cross-fertilized them. It didn't really matter. And what did he get? Well, let's do a Punnett square. If you know that you're breeding, if a heterozygote tall plant can only make two types of gametes, either a big T gamete or a little t gamete. A heterozygote mom plant can only make two types of eggs, gametes in this case, one sperm, one's eggs, a big T and little t. Well, what did he get for babies? Whoops. Conventionally, we usually write the capital letter first. It doesn't really matter. But by convention, we usually just write the capital letter first. Well, phenotypically, we got 75% tall and 25% dwarf. Phenotypically, 75 or 3/4 tall and 25% or 1/4 short, dwarf. That's what we got phenotypically. Genotypically, we got a 1 to 2 to 1. We got one homozygous dominant tall. We got two heterozygotes, and we got one homozygous recessive short. All right, that's what we got for our F2 generation. So our F2 seeds would have been, all right, and F2 plants, tall, short. So that was a single factor cross. Shows simple dominant recessive relationships. This, by the way, not only showed law of segregation and the dihybrid cross showing law of independent assortment, but it also showed that the unit of heredity is particulate. It's not blended. It's not cut up and shuffled. that the gene is a particulate in nature. And we know now that genes, he didn't know this at the time, but we know now that genes are a piece of the DNA and they are particulate in nature. And by the way, I'm talking like these ratios are perfect. Look at the 3-to-1 ratio here. It's really 3.15 to 1. And this is really what he got for raw data, 705 to 224, 651 to 207. Remember, it depends on how many seeds you plant. If you plant 100 seeds, you might be a little further from the 3-to-1 ratio. If you planted 1 million seeds, you're going to more closely approximate or approach that 3-to-1 ratio. So you can see it's really close to a 3-to-1 ratio, 2.96 to 1, 2.95 to 1, 2.82 to 1, 2.84 to 1. And in fact, that's why we're going to do the chi-squared analysis later. Later on, I'm going to teach you that the chi-squared analysis is the observed minus expected squared divided by the expected, and you sum all of those. And I'm going to teach you how to do the chi-squared analysis later. What we're really saying is 3.01 to 1 the same as 3-to-1, or is it significantly different? Well, the chi-squared analysis is analysis of this variance, and it tells us, is 3.01 to 1 really 3-to-1, or is it significantly different than 3-to-1? For example, is 3-to-1 the same as 6-to-1? I think intuitively you would say no way. That's significantly different. But let me ask you this, is 3-to-1 the same as 2.9999998 to 1? And I think you would intuitively say, boy, that's really close, that's the same. But we can't just say intuitively in science that's the same. We have to show it's the same. And later on, we're going to do a chi-squared analysis to show that this is the same. All right, from all of his experiments with his monohybrid cross and his dihybrid cross, he said that that there's discrete units of heredity called genes. You get one from your mom and one from your dad. He said that you could be homozygous, like big T, or little t. He said that you could be heterozygous, big T, little t. So he said this. He postulated that. He with the alleles he was looking at, the traits he was looking at, he had simple dominant recessive relationships. Tall was dominant over short. Yellow was dominant over green, etc., etc. So he saw the simple dominant recessive relationship. And during his experiment, he said that your genes, your homologous genes, the gene from your mom and the gene from your dad, separate. Your alleles separate. And he called that the law of segregation. All right, so let's talk about some terminology now. Gene is the unit of heredity. It's Mendel called it a unit of heredity. We know now that it's a piece of DNA. That's what we know now. Alleles are variants of the gene. Remember, a characteristic is like height, but the traits are tall and short. Well, alleles are the variations on the gene, and the gene codes for that characteristic. So there's a gene that codes for height, but the allele is the tall gene and the allele is the short gene. Homozygous means that your alleles are the same, and heterozygous means that your alleles are different. Genotype is what your alleles are, and phenotype is what you look like externally. All right, we've done enough Punnett squares here. I don't think I have to. There's no rule saying you have to put the sperm on top and the eggs along the side. You could switch that, but you should probably stick to whatever you do, whatever you like doing. And maybe if you've never done this before, make sure do what the book does. Put your female gametes across the left. Put your male gametes across the top. If you always do the same thing, it's just a little bit easier. But there's no hard, you don't have to. You get the same results if you switch those. All right, the law of independent assortment. This came from the dihybrid cross. So your book's calling it a two-factor cross, but this was the dihybrid cross. This is where we crossed a big R, little r with a big Y, little y. So we crossed a round yellow seed, a plant that made a round yellow seed with another plant that made a round yellow seed. That's what we did. And we looked at what came out. And if you remember phenotypically, what came out was 9 to 3 to 3 to 1. This would not have come out that way if these weren't on separate chromosomes, and if how chromosome number 1 homolog separated wasn't independent of how chromosome number 2 homolog separated. Let's take a look at that. Okay, before I go on, I want you to notice something. Because it's finally using it. When you have a 9 to 3 to 3 to 1 ratio, you know your frequencies. You have 9/16. 9 plus 3 is 12, plus 3 is 15, plus 1 is 16. If you add those together, you get 16. So 9/16 of your offspring are round in yellow. 3/16 of your offspring are wrinkled in yellow. And 3/16 of your offspring are round and green. And only 1/16 of your offspring are green and wrinkled. So this is what that's showing you. Well, I didn't finish that. Let me finish this for you. Oh, it wants me to do it. All right. Yellow or green. I'm just reading it for you, that's all. So what we're saying here is this. This is not too bad. If I have 75% chance of being yellow and 25% chance of being green, then what is my percent chance of being yellow and round? Well, it's 0.75 times 0.75, because I have 3/4 chance of being yellow and I have 3/4 chance of being round. So what is my chance of being yellow and round? 0.75 times 0.75, and I'm just going to do that in the calculator right here. 0.75 times 0.75 equals 0.5625. So it's not letting me write anything in there, but it would be 0.5625. And what are my chances of being yellow and wrinkled? Well, I have 0.75 chance of being yellow and 0.25 chance of being wrinkled. So let me just do that. 0.75 times 0.25 equals 0.1875. I have 0.1875 chance of being yellow and wrinkled. And I won't keep going. But all you have to do is multiply these probabilities together. That's what you have to do. And I think I told you that to be yellow and round was 0.5625, and what that is 0.75 times 0.75. And indeed, if you do 9 divided by 16, you get 0.5625. So this is just the frequencies or the probabilities of getting these genotypes. All right. This is just, okay, this is an experiment done. What he says was, what if the R and Y are linked? What if big R is always with big Y and little r is always with little y? Well, if that's the case, then you can't get 4 gametes. This parent can only give you one type of gamete. All right, all it can give you is one type of gamete, big R, big Y. And this parent can only give you one type of gamete, little r, little y. And you mate them together and you get a heterozygote. But remember, the big R and big Y is still linked. They're on the same chromosome. So this dihybrid crossing and big R, little r with a big Y, little y, you can still only get 2 types of gametes. You're still only going to get the big R, big Y, or the little r, little y, because the big R is on the same chromosome as the big Y, and the little r is on the same chromosome as the little y. This could be a possibility. And likewise, well, not likewise, but conversely, you have the other hypothesis that says the big R and the big Y are on different chromosomes. And if they're on different chromosomes, then you can get these different gametes, 4 types of gametes, 25% chance of getting each of those. Well, if I was doing a cross Oh, actually, I think I left a slide for myself to do that. So let me not do that on this slide. I'll run out of room. So those are my two hypotheses. Good. This is I left myself room to do this. So if I was doing that, well, I only make two types of gametes. This parent right here can make a big R, big Y, and it can make a little r, little y. How come the big R can't go with a little y? Because they're linked. See, you can't have the big R with the little y. Now, you could if they're on separate chromosomes because they're going to independently assort, but you can't because they're on the same chromosomes. And likewise, the other parent, because the parents are the same, genetically the same, the other parent can only make two types of gametes. So is this what we found? Did we find kids that were round and yellow, round and yellow, round and yellow, and wrinkled and green? And you're going to say to me, well, we found some kids that were round and yellow and some kids that were wrinkled and green. Yeah, but where's my wrinkled and yellow? Where's my round and green? Where are those? Did we find this? And did we find it in this proportion? And the answer is no, we did not. We did not find this distribution of progeny when we did the dihybrid cross. When we did the dihybrid cross, we did not find this distribution. What we found was that they were on separate chromosomes, and we could make four types of gametes. And the four types of gametes we could make was big R, big Y, big R, little r, big Y, and little r, little y. And likewise with the mom. That's what we found right here. Little r, big Y, little r, little y. And then I'm not going to fill these in, but you could fill these in, and you could see that you're going to get your 9:3 to 3:1 ratio. So that's what we found, and that supported our independent assortment that the R and Y are in separate chromosomes. This is using S and Y. Oh, smooth, yeah. Remember how I told you that some experiments don't use R for round, they use S for smooth, but you got to be careful because the pods are also either constricted or smooth. Well, now you're finding a source that uses smooth instead of R. It's not different, folks. It's just not different. And then you plant them and they grow, and you cross them, and this is nothing different than we've talked about the whole time. If you don't have it, and I think you probably do, because it's really not that bad. It's just bookkeeping, keeping everything in its place. But if you don't have it, go back and play the videos again. Remember, the fact that you're using S for smooth instead of R for round, it doesn't matter. You just got to realize that the nomenclature is a little bit different. I'm going to let this play out and show you the 9:3 to 3:1 ratio. There will be 9 smooth yellow. There will be 3 smooth green, and there will be 3 wrinkled yellow, and there will be 1 wrinkled green. That's a 9:3 to 3:1 ratio. There you go. There's your 9:3 to 3:1 ratio. All right. Oh, let me go back and show you something. There's a simulation part of this, and I can do some mating. And you don't have to do this, but you can look at the peas themselves, and I can say produce 1 offspring, and or I could say produce 1,000 offspring. There's your 9:3 to 3:1 ratio. Produce 1,000 more. 1,000 more. Oh, it's not actually adding up. It's 1,000 each time. So you're going to say to me, Kiggins, this is 7.9. It's not 9. Well, I only did 1,000 offspring. If I would have done 10,000 offspring, it'd be closer to 9:3 to 3:1. Remember, it wasn't exact, but you could do a chi-squared and see if it's different. You can also go look at the plant itself, and you can do some. This is going to be tall with purple, or short and white, or tall white, or short purple. And I can produce 1,000 offspring, and I get my 9:3 to 3:1 ratio. All right, so there's a simulation part of this with the principles. All right, we're getting there. Now, when I do this dihybrid cross, I have, if you look at my F2 generation came from selfing my F1 generation, which was dihybrid. And if you look at this, my non-parentals are novel. Anything that's not round in yellow is a non-parental, 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 non-parental. I missed one. Non-parental right there. These are non-parentals. And it should be 7. It should be 9 and 7. It should be 9 to 7. 9 parentals with 7 non-parentals. 1, 2, 3, 4, 5, 6. Yeah, that's 7. You could even look back at the parental generation, the grandparents, and say who's non-parental. Well, there's 6 non-parentals, because guess what? That one's parental. You could say, well, that's grandparental. Oh, okay, that's grandparental. But really what we started with was round yellow, wrinkled green. So now I have these 6. And by the way, if I'm asking you a question and if you're saying, are you talking about the true parents, the F1, who are the parents of the F2? Or are you talking about the grandparents? Call me over to your table, grab my tie, pull me down, and say, Kiggins, I need to know if you mean the grandparents or the parents. It's a fair question. So the non-parentals here, if I'm really talking about the grandparents, are the green smooth and the wrinkled yellow. Because who did we have to start with? We had smooth yellow and wrinkled green. So you can see that these novel combinations, or what we call non-parentals. And by the way, the non-parentals are called recombinant types. Now you might think recombinant DNA is sewing 2 different species DNA together, and you'd be right. But recombinant DNA is actually putting DNA together in new combinations as well. New combinations like wrinkled yellow. There's no wrinkled yellow combination here. There's no wrinkled yellow combination there. There's no wrinkled yellow combination there. So I got a recombinant combination, a recombined combination when I have wrinkled yellow. That's called recombinant types. All right. When you're looking at the phenotypes of, I don't know what this is really trying to, it's showing you the 9 to 3 to 3 to 1 ratio. That's what it's showing you. It's showing you 2 different ways of doing it. It's showing that you can do this line method or you can do the Punnett square. That's pretty simple. You can do it either way. You're going to get a 9 to 3 to 3 to 1 either way. So you can draw out the Punnett square. You can do the line method. I will tell you that the line method is easier for statistics. Definitely statistically it's easier to use the line method. I'll show you an example. But whatever way works for you, you could do the Punnett square or that. This is showing you, by the way, you can do 3 fractor crosses, a tri-hybrid cross. You're going to say, wow, don't make me. I'm not. It would be an 8 by 8 Punnett square, a 64 box Punnett square, 8 by 8. And how do I know it's 8 by 8? Well, if you're doing a tri-hybrid cross, well, I'm going to have 3 different types of alleles, and I'm going to have, for this spot right here, I'm going to have yeah, that's right. That's completely right. I'm going to have 2 choices. I can put a P or a Q there. Oh, by the way, what is P plus Q? Well, let me just tell you what that is. P plus Q cubed is really P plus Q times P plus Q times P plus Q. That's really what it is. You're going to say, how come P and Q is used for my T, my R, and my Y? Well, P plus Q is really the frequencies. It can be the frequency of the T, the frequency of the R, and the frequency of the Y. But just go with it for now, P plus Q. That's P plus Q to the cubed. I have 2 choices to go in this slot right here, P or Q. I have 2 choices to go in that slot right there, P or Q. See, I have a slot right here, 2 choices. I have a slot right here, 2 choices. I have a slot right here, Two choices. Well, how many different combinations do I have with p plus q cubed? And it's 2 times 2 times 2. 2 times 2 is 4 times 2 is 8. That equals 8. I'm going to have 8 possibilities of gametes. But the male is going to have 8 possibilities, and the female is going to have 8 possibilities. So my Punnett square is going to be 8 by 8, or 64. That's what it's going to be. 64 box Punnett square. And all this is saying is, here what you do is, first you take your tall and your dwarf, and you multiply it. Well, really, this is what you're multiplying. Tall plus dwarf times round plus wrinkled times yellow plus green. But first, let's keep it simple. First, do the tall and the dwarf times the wrinkled, the round and the wrinkled. First, do that. And then here's what you get. Here's what you get. All right. And then you take that and you multiply it by your yellow and your green. And here's what you get. You're going to say, well, that gets kind of daunting. Well, it does get kind of daunting. Luckily for you, we're going to do a binomial expansion later. Let me see. Let me just do this for you. I'm running out of time here. Let me do this for you. If I want to know how many out of 64 is capital A, capital B, capital C, all I got to do is, well, I got 3/4 of a chance of being capital A, and I got 1/4 chance of being little a. So this is 0.75, or 3/4 times 3/4 times 3/4. I got 3/4 chance of being big B. So 3 times 3 is 9 times 3 is 27. Oh, I think I have to type in there. Let me see. Yes, 27/64 chance of being capital A, capital B, capital C. What are my chances of being capital A, capital B, little c? Well, 3 times 3 is 9 times 1 is 9. And by the way, 4 times 4 is 16 times 4 is 64. That's how they're getting the 64, so that's 9. What are my chances of being A, little b, c? Well, 3 times 1 is 3 times 3 is 9. 9. What are my chances of being A, little b, little c? Well, 3 times 1 is 1. I'm sorry, 3 times 1 is not 1. 3 times 1 is 3 times 1 is 3. 3. What are the chances of being, you got it, 1 times 3 is 3 times 3 is 9. What are the chances down here? 1 times 3 is 3 times 1 is 3. What are the chances here? 1 times 1 is 1 times 3 is 3. What are the chances here? 1 times 1 is 1 times 1 is 1. I hope this is right. They're all right. Okay, this is an easier way to do it than doing an 8 by 8 or 64 box Punnett square. It's definitely easier doing it this way. And that just shows you that. Oh, that's the answers. Oh, that's why I did this first so you could see it done out manually, and then here's your answers right there. All right. All of these tenets of Mendelian genetics led to this chromosome theory of inheritance, but Mendel himself did not know these genes were on the chromosomes. But he did know the gene was a discrete unit of heredity. And later on, we came up with genes are on chromosomes. So it came up with this chromosome theory of inheritance. All right, so what led to the chromosome theory of inheritance, if all where Mendel stopped was genes are the unit of heredity? That's where Mendel stopped. Well, some people added into that. Mendelian, Weissman, Bovarian, Sutton, Strasburger, Fleming, not Sir Alexander Fleming, but Walter Fleming, I believe. There's a couple Flemings. Just so you know, there's a couple Flemings, and I think it was Walter Fleming. So first of all, we start with Mendel's breeding experiments, and then we looked at a couple other things. We say that there's this stuff in the nucleus called nuclein. And the nuclein seems to be involved in your inheritance, so we see that. So then we suggest that chromosomes carry genetic material. But chromosomes are made of two things. They're made of DNA and protein. So what's the genetic material, DNA or protein? Well, all right, we don't know yet. Well, we know now, but we didn't know back then. We also saw that the chromosomes in the nucleus, they separate during meiosis. Well, Mendel knew that homologs separate. He didn't know that the homologous genes, the alleles, he didn't know that the alleles were homologous genes on homologous chromosomes, but he knew that homologous genes, i.e. alleles, separate. Well, we saw these separating during meiosis. So with Mendelian, Weissman, and Fleming, Walter Fleming, and Bovarian, Sutton, with a whole bunch of other scientists adding to what Mendel already have already stated, we came up with our chromosome theory of inheritance. We know that the law of segregation can be explained by the separation of homologs. Mendel said that alleles separate, but we know that alleles are just homologous genes. We said that the law of independent assortment is explained by the random alignment of the homologs at the metaphase plate. Mendel didn't know there was a metaphase plate. He didn't know the tetrads aligned at the metaphase plate. But he did know that these homologous genes, these alleles, independently assorted from each other. So the big Y, little y, independently assorted from the big R, little r. They didn't, how one assorted didn't affect how the other sorted. So that's explained by the random alignment during metaphase 1 of meiosis 1. That's what explains that. All right, so now we take all of this and we start studying human genetics. Well, here's the problem. It is unethical to tell a lady that she has to go out there and have 27 kids for you to do some genetic analysis. 27 kids, even back in the day of a rural farm society, I mean, they had big families, 12, 18. I know a family from my hometown that had 18 kids. So that was back in the 70s and 80s, 18 kids going to school, but you don't see that anymore. And even 18 is not enough for genetic analysis. So what do we do with humans? We do pedigrees. We do a pedigree analysis. We trace the genes through a family. We put the Roman numeral on the generation that we start with, Roman numeral 1, Roman numeral 2 generation, Roman numeral 3 generation. The proband is whoever reports. This could be the proband. The proband is whoever reports to the genetic counselor. But the genetic counselor then puts together your pedigree. Boxes are males, circles are females. Half filled in are carriers. They don't have the disease, but they're known to be carrying the disease. Fully filled in is diseased or trait of interest. I mean, it doesn't have to be a horrible disease. It doesn't have to be a disease that kills you. It could be color blindness. Just disease of interest, just a trait of interest. So disease or trait of interest. So you got carriers, you got disease, you got females, you got males, you got generations. You can see how we write dizygotic twins. I don't have one over here, but right here, you can see how I write dizygotic twins. Dizygotic twins are fraternal twins, non-identical twins. They came from two zygotes that fused. I'm sorry, they didn't fuse. Jeez, that's a tetragrammatic chimera. They came from two zygotes, two different eggs, two different sperm. So two different eggs, two different sperm. Dizygotic twins, non-identical twins. Identical twins, you can see, are written similarly, but we connect them across the center. We say they're identical twins by connecting them across the center. So this is a pedigree. You do need to know about pedigrees. You absolutely need to know about it. So I would commit some of these conventions to memory. All right, I'm stopping here. I'm going to make another lecture on probability and statistics. That is probably going to be every bit as long as the rest of Mendelian genetics, but it's going to be math. So, and you can listen to it several times. So I'm going to stop right here and do another lecture. See you in a few, folks.